Okay, good afternoon, everyone. We will continue our council seminar series today with a presentation by Dr. Tarik El-Mellouki. So his presentation is going to be about mining microbial biosynthetic gene clusters. Tarik, you can go ahead and start. Thank you very much.
Thank you, Mulay Abdelmajid. So my name, as Mulay Abdelmajid mentioned, is Tarik El-Mellouki. And my presentation today is going to be about mining microbial biosynthetic gene clusters. And I think that this is going to be a 21st century gold rush. And I'm going to try to convince you through the upcoming slides. I'm going to tell you a little bit about myself before I get into the subject itself. So I come from the city of Rabat in Morocco. That's where I fell in biology. That's where I did a bachelor's in plant biology. And then I went to France, to Paris 11 University, where I did a master's degree in plant developmental biology, and I worked at Institute of Biotechnologie des Plantes in Saclay, France. And after that, I moved to Southern Illinois University at Carbondale, where I worked with Dr. Khalid Mixen, and I worked on tilling, which is a functional genomics tool in soybeans. And after that, I moved to University of Missouri, Kansas City, where I prepared my Ph.D. And since I graduated with my Ph.D., I've been teaching, and I've been teaching at a small community college called Johnson County Community College. I've been teaching different courses in cell biology and molecular biology, but also in microbiology. And recently, I've joined Catalan's Pharmaceuticals as a scientist in their preclinical studies. So before I get into the presentation itself, I would like to put some references here for you. So if you want to go back later on and find some of the references that I'm using in this talk, you can go back to this slide. All right. So my talk outline is going to be as follows. First, I'm going to talk a little bit about microbial diversity. What is the current assessment? Then I'll talk about biosynthetic gene clusters, try to define them and describe them. We'll talk briefly about some newly discovered BGCs, some examples that are relevant. And then we'll talk about precision medicine and the quest to drug the undruggable, and then the role of synthetic biology in exploiting microbial metabolism. And in this, we'll talk about the design, build, test workflow in synthetic biology. I'm going to briefly talk about design, build, and test, and then the role of artificial intelligence and machine learning in that. It's not going to be anything extensive. I'm going to just give you an overview. So this is more for students to understand what's coming up as a science. So if as they prepare for graduate schools, they can keep these things in mind if they are interested. And finally, I will talk about a program that I am part of called Tiny Earth. And it's one of the tools that can be used to access biodiversity in the classroom. So let's get started with microbial diversity. And one of the things that I always tell my students, and I'm going to use some of the slides I use with my students, is that microbes are highly diverse. And this is something evident. We know that microbes can be either prokaryotic, and these are organisms usually or are unicellular, where the DNA is not enclosed within the nucleus. And in prokaryotes, of course, you find the domain bacteria, the domain archaea. And then also we have eukaryotes. So there are microbes that are eukaryotic, and these are organisms that contain DNA within a nucleus. To simplify, there are other characteristics, of course, but just to simplify the difference between prokaryotes and eukaryotes. And within the eukaryotes, you're going to find protists, which are mainly unicellular. You're going to have plants, fungi, animals. And as I said, these organisms are mainly characterized by cells that are larger and contain DNA within the nucleus. And then finally, we have viruses. So these are neither eukaryotes or prokaryotes. And these are organisms that require infection of cell in order to reproduce. So this is just to give you a little bit of an overview. What are microbes? And so as you can see, they are highly diverse. In recent work, and especially with the advancement in sequencing techniques and sequencing technology, we have unraveled a hidden diversity. So if you recall, or if you probably know this, the early studies of microbes relied on culture-based studies. Basically, you had to isolate the microbe, and you need to be able to culture it in order to study it. Nowadays, we don't need to, let me digress a little bit. We don't have all the tools to grow all the microbes. There is an estimate that only about 1 to 2% of all microbes can be grown in the lab, especially in bacteria. So when you take a sample, an environmental sample, for example, and you put it on a growth plate in a medium, you only see a very, very small fraction of all microbes. So how can we access the hidden world of microbes? Well, one of the tools is sequencing. Basically, this doesn't require to culture the bacteria, and the advancement in metagenomics have revealed that there is an extensive biodiversity that we were not able to see before, that we now can see with the identification of sequencing. So basically what we can do is get a sample from an environment and then extract all the DNA from the environmental sample, and then subjugate this to extensive sequencing or deep sequencing, and then reassemble genomes based on different programs, which I'm not going to get into here. And then that can yield or manifest a lot of novel species. So in recent work, especially this one that I'm showing you here, a new view of the tree of life that was published in 2016, this group proposed novel taxa, new taxa that were not identified previously. Now, from this slide or from this image, this is a phylogenetic tree, if you will, cladogram, and you can see that there are multiple branches. And if you will, the take-home message from this image is that the domain bacteria, which I highlighted here, contains the largest number of species in comparison to archaea or eukaryotes. The biodiversity of the microbial world is particularly evident in its metabolic versatility. So microbes are extremely diverse, and their diversity is not in, they have a lot of forms and shapes, but if you will, the area where there is a lot of diversity in the microbial world is metabolic activity. In fact, all multicellular and non-microbial organisms depend on the metabolic activities of microorganisms. You, me, other eukaryotic organisms, we depend on microbial species. So let's talk a little bit about metabolism. So there are two types of primary metabolism in cells, either primary or secondary, right? Primary metabolism is required for growth, development, and reproduction of the organism. This is essential. Primary metabolites are going to be produced by many unrelated taxa. They're going to be found in different species because they are essential for the function of the cell. On the other hand, secondary metabolites are organic or defined as organic compounds that are produced by bacteria, fungi, or plants, which are not directly involved in the normal growth, development, or reproduction of the organism. So what do they do? Their role is to enhance the survival of the organism producing them. This, for example, could be antibiotics produced by bacteria and fungi to compete for a niche or an environment in which they are growing. Most secondary metabolites are synthesized by enzymes that are encoded within what we call biosynthetic gene clusters. So what are biosynthetic gene clusters? They are defined as locally clustered group of 2 or more genes that together encode a biosynthetic pathway for the production of a secondary metabolite. For example, what you will find in a biosynthetic gene cluster are chemically defining synthases or synthetases. So these go in to take primary metabolites and going to transform them and make a carbon backbone. Additionally, what you will find in all canonical biosynthetic gene clusters or BGCs, what we call tailoring enzymes. Those are particularly important, as I can talk about later. But what do these tailoring enzymes do? They are going to further modify the carbon backbone of the secondary metabolite. They might add or remove a functional group that might render that secondary metabolite active or inactive, depending on the downstream activity. These biosynthetic gene clusters, all of them in general, not all of them, but many of them can have a unique transcription factor, just like the case of operons in bacteria, where you have one promoter controlling the expression of multiple genes. These BGCs can also have a central control unit that can control their expression, either to turn them on or off, and so on. But what's interesting is a lot of these BGCs also, or some of them, contain genes that will express what we call decoy molecules. So if you can imagine if a microbe is producing secondary metabolites with an antimicrobial activity, that particular microbe doesn't want to be affected by that secondary metabolite. What usually they do is they will express a protein or some molecule that will limit or inhibit the effect of the secondary metabolites on the producing organism itself. And finally, in a biosynthetic gene cluster, there are some other genes that can be found based on all the genomic data analysis. And so a lot of them are still unknown with not known function. So the bottom line is that you have cluster genes controlled sometimes with one single transcription unit. And these can be either ones that are going to make the backbone of the secondary metabolites and what we call tailoring enzymes. The others can be accessory and depending on the organism producing. Right, let's see. So what are the different classes that are produced, or different classes of secondary metabolites produced by BGCs? So in general, and this is not exhaustive, they can either be derived from acetyl-CoA or they can be derived from amino acids. Those that are derived from acetyl-CoA can be polyketides, such as lovastatin. And you probably know this is a cholesterol-lowering drug that is being used extensively in the biopharmaceutical industry. Other molecules derived from acetyl-CoA that are secondary metabolites are terpenes. And so these go through a mevalonate pathway intermediates before you can produce these terpenes. So there's a large number of them. I'm not going to go into any specific one of them. And then the ones derived from amino acids, those are peptides. And what's interesting is that some of them what we call non-ribosomal peptides. So for students amongst us, the traditional way we understand about peptide production is that all produced by ribosome, gene expression. But there are some other peptides that we call non-ribosomal peptides that are not produced directly by ribosome. So these are the general classes, but you can have hybrid molecules. You can have molecules that have polyketides and terpenes. You can have secondary metabolites where you have, for example, you can have a non-ribosomal peptide with a polyketide, or you can have a polyketide with a fatty acid. So there's a huge variety of the different combinations that can be produced by microbes. Now, the title of this presentation is Mining Biosynthetic Gene Clusters. And more data is being produced to highlight that there is a great diversity in terms of the type of secondary metabolites that are produced by different taxa. Perhaps one of the famous taxa called actinobacteria. These are generally associated with antibiotic production. And you can see here in this blow up or the image is that they contain all different classes of biosynthetic gene clusters or secondary metabolites. But there are other ones. And the more sequencing we have, the more we are going to unravel of this diversity. So here, for example, the Pseudomonas also have a large diversity in terms of secondary metabolites. The Burkholderia also great diversity of biosynthetic gene clusters. So this is a very important and very interesting. Why? Well, biosynthetic gene clusters or secondary metabolites in nature have different roles. They can allow for microbial communication, either in symbiotic or commensal interactions. And this is going to be, we can exploit this. So for example, you can imagine if you can unravel the type of molecules involved in communications between a plant and microbe, you could, engineer or you can interfere with these interactions either to benefit the plant or to prevent the plant from being harmed by a specific microbe. They are important for development, reproduction, mating. And so if you have microbes that are pathogenic, we can try to control these pathogenic microbes. But also if you have microbes that are beneficial, we can also interfere with the development if we can understand more about these secondary metabolites. These also protect the microbes from self-produced toxic compounds, as I mentioned earlier, so the production of decoy molecules. And these are particularly interesting because a lot of these decoy molecules can be conserved proteins, for example, that can exist in humans. And so you can imagine that because these decoy proteins are shared between, for example, fungi or bacteria and humans, these could be targets for drugs. A lot of these secondary metabolites are used by the microbes to defend themselves against invaders. For example, a fungus against bacteria. So here is the example of Fusarium species that is protecting itself from a bacterial virus called ralsolamycin. And then, so if you don't have the BGC in this particular species or Fusarium species, then the bacteria are able to grow and invade the cells and cause harm to this fungus. But if the BGC that is called bicaryarian BGC is present, this can halt the growth of the bacteria and prevent the harm to the microbe. So a lot of antibiotics that we use are derived from fungi, and for this specific reason, and because we share a lot of the biology with fungi, we can use these antibiotics. One of the key things about antibiotics is that they need to be have low toxicity for humans. So a good antibiotic is an antibiotic that targets something that doesn't exist in the human cell. So that way it will specifically target the bacterial invader, for example. What's interesting is that these BGCs can be shared. So the BGC for bicaryarian is found in Botrytis cinerea, and then it was transferred to Fusarium through horizontal gene transfer. So it is very interesting to look at also these horizontal gene transfers to understand how these BGCs can evolve, because if we understand how BGCs evolve, then we can discover de novo BGCs or BGCs that are not readily discoverable using traditional bioinformatic tools. Right now, most of BGCs that are discovered through homology. Basically, you look for things that resemble previously described BGCs and you try to characterize them in novel or newly sequenced organisms. One of the challenges is how to discover BGCs without homology. And so there is a lot of work that is being done right now to write algorithms that look for specific molecular signatures or sequence signatures that can allow for this type of discovery. All right, let's look now about some of the newly discovered BGCs. And I'm just going to give you two. There's a lot. What you need to know is that we know that BGCs are highly abundant in nature, and especially in commensal associations. In the fungal kingdom alone, new algorithms, even with conservative predictions, estimate that there are several million unique biosynthetic gene clusters. That's a very large number with a large potential in discovering new molecules and new molecular signatures. So many members. This was interesting to me also as I was teaching microbiology and I'm looking for new discoveries. One of the discoveries that struck me since I was talking about antibiotics to students was this paper in Nature that discovered that some of commensal bacteria can produce antibiotics that actually target MRSA, target methicillin-resistant Staphylococcus aureus. So within our bodies, either on our skins or within our guts, we know for a fact that there are microbial species, either bacterial or fungal, that are able to produce chemicals that are beneficial to us. And this is one of the things that are extremely important for us as a human society. As we change our behaviors, there are studies that show that the biodiversity in the human gut, for example, is reduced as we move into a more Western diet, if you will, rich in carbohydrates, less diverse, less using less complex sugars, for example. And then so one of the challenges, and there is a lot of research going on right now with microbiomes, is to try to understand how microbiomes evolve to conserve our microbiomes, to conserve the richness that we might have, especially for us, for example, people who've lived in Africa, for example, lived in environments that were less westernized. Those people probably have a lot of diversity in their microbiomes than Western societies or Western style societies do have. Anyways, that was just a side note. But the discovery found that there is this species, commensal species, that is able. So here in this image from the paper, let's see if I can find mine. Yeah, so here, this is a lawn of bacteria. So S. aureus, the MRSA, for example, spread on the plate. And these are the new species that are isolated. And so this is the wild type version. It can create this zone of inhibition, preventing the growth of S. aureus. The group did some genetic manipulations to knock out the genes responsible for the production of the secondary metabolites responsible for this zone of inhibitions. And then eventually confirmed the biosynthetic gene cluster by reintroducing it into this species. So the discovery led to the identification of a new novel biosynthetic gene cluster that is being studied, and I think it is going into preclinical or clinical studies. I'm not sure, but it is a promising new antibiotic, because you should know that right now we are facing an antimicrobial crisis. A lot of bacteria are developing resistance to antibiotics, and we are running out of antibiotics. It's a serious threat, and it is being classified by CDC or by World Health Organization as one of the 21st century threats to human health. This is just another two examples. So here in 2019, the identification of the bacterial biosynthetic gene clusters. This is for oral microbiome. And this was interesting because they discovered some novel molecules that allow the microbiome to communicate in the oral cavity. What's also very interesting is some environmental places that have not been well exploited and well researched are aquatic environments. And the ocean, for example, still contains a lot of mysteries in terms of biodiversity, fungal and bacterial biodiversity. So there's a lot of research now focusing towards these novel environments that haven't been exploited before. One, because either they were not accessible, or two, because we didn't have the tools to grow the microbes, and now we can sequence. So what a lot of people are being able to do is basically they can sequence the genomic, the genomic DNA, and they can unravel the BGCs, and they can take the BGC sequences and put them in a different species and try to express them if the bacterial species or the fungal species from which they were discovered, if it's difficult to grow or if it's difficult to manipulate in the lab, we can use more familiar you know, species that we use in the lab and transfer those BGCs. So there's a lot of work now to try to engineer strains with heterologous expression cassettes coming from other organisms. All right, so in the next few slides, I'm gonna just summarize a little bit what's the challenge and why BGCs could be the next place to mine for drugs for humans. And so you possibly have heard about precision medicine or the undruggable. So in the future, we want to tailor the medicine to specific patients, but also one of the problems right now is that there are many targets that are undruggable. Basically, we're not able to find drugs for them because of different reasons. And so microbes, because of this extensive biosynthetic ability, can be used to define or to find novel drug modalities. So as I said, the broad problem is too high to identify targets. So basically, what is the drug is going to target in the human cell, and how to target them. Can we get the drug to these targets? Because as you can imagine, in order for a drug to work, it has to cross several barriers to get inside of the cell. And once inside of the cell, it has to find its target. So there are two major categories of drugs that are used in the pharmaceutical world. Biologics, these are the more recent ones. So these are very large molecules that have large contact surface areas. So because they have a large surface areas, they have great stickiness. Basically, you have a good ability to bind to the target. But the problem, because they are big, they hard to cross the cell membrane. So there are different tools that are developed to allow these biologics to reach inside of the cell. And then the other category are small molecules. So these are the older family or type of drugs that are used. And these have the advantage of being small. They can cross plasma membrane, but the fact that they are small they have limited adhesive power. So it's very difficult for them to stick to the target. And so it can take a very long time to screen through different small molecules that have a high affinity for the target itself. What's interesting is that both these drugs in the market or being developed right now only target about 20% of all targets in the human cell. So the vast majority of human proteins are not targetable; they're not druggable. So you can imagine. Let me just read this. It has been estimated that as many as 3,000 genes encoding proteins can have their activity modified by pharmaceuticals. However, only 5–10% of what is considered druggable is currently targeted by FDA-approved drugs, reflecting opportunities for further research. So this is from Eliminating the Druggable Genome in an NIH program, if you are interested to read more about it. But again, so this is opportunity, and this takes me to my second sentence in the title, which is the gold rush. So even though only 10% of all proteins are druggable, the annual revenue between the biopharma and the biotech industry is estimated to about $750 billion and $250 billion respectively. Just to give you a point of comparison, Moroccan GDP is about $120 billion. So just from 10% or 20% of the protein is targeted, it is a lot of money that is generated. And if we can access another 20%, that's a niche, that's a gold rush. There is opportunity there. There is the ability to discover something novel or discover many novel drugs that can be used to target undruggable molecules. What's interesting is that, I mean, this is something that is evident: about a third of all medicines used in the clinic are derived from microbes and plants. And so the ability to exploit novel biosynthetic gene clusters will allow to identify new drug modalities in the future. So can we identify novel drugs modalities? The answer is yes, very likely, especially now with novel approaches to drug discovery, especially the use of microbes and use of synthetic biology. All right, so this takes me to the next part of my talk, which is a little bit about synthetic biology. I'm not going to go into any details, but this is a field that is poised to transform drug discovery in the future. The development of drugs, this is just to remind you about the steps that go into developing a new drug and have it approved for use by patients. The first stage, which takes the longest time and perhaps the largest amount of experimental approaches, is drug discovery. So during drug discovery, you have to identify a large number of compounds, and then from those compounds, you narrow it down to during preclinical development. This is when you look for potency of the drug, efficiency of the drug, stability of the drug, and so on. And then after that, from those about 250 compounds, we move to a smaller amount, which is 5 compounds that are used in clinical studies. And this when you start to test the drug in animals and also, in humans, and you go through the different stages of phases in clinical development. And then finally, the final stage in drug development is a regulatory approval. So once you have all the data, you have to submit it to a regulatory organization, for example, the FDA, in order to get approval for the drug. So you can see the amount of work that is going on. And if you will, the limiting step is the first step. You need to have good candidates in order to get to the one drug that will work. So drug discovery involves, so this first step here is going to involve two things. One, you have to discover a target. So basic research that is done in different universities, different labs. As we understand more about how the cell works, then we can discover new targets. So the more we understand the biology of the cell, the more targets we can identify. And then the second stage of drug discovery is finding a drug candidate. Target discovery must pass through a step called target validation, which also during drug discovery. And then drug discovery requires hit generation. Basically, you need a molecule that binds your target. And then out of those molecules that bind the target, you need the lead candidates that can be used later on. So what's important right now, or I guess it's a game-changing approach, is that the preclinical work can be enhanced using strain engineering, basically making strains that produce either human targets or human drug candidates, and automation. So because, as you said, if you're doing screening for drugs, you need thousands and thousands of screens. And so automation is essential. So this is a field that is growing extensively. And here is my pitch for students who are looking for opportunities. Automation in biological labs and research is one of the fields that is hiring more and more, or looking for more and more students. And finally, machine learning to speed up this whole process. These are things that are already and are going to become more and more instrumental in the process of drug discovery. Strain engineering goes through several stages, and so this is something that is extremely important if you want to use synthetic biology. You have to go through a step of design, and the design can be nowadays aided by computer software. So basically, instead of designing by hand, cloning steps. You can all do this automatically and have a robot handling your cloning and all this stuff. The build, so basically you have to get the strain built, and then you have to test that strain. And so usually now what they do is they test thousands and thousands of strains in automated processes. All of this require different approaches. You need genome mining and bioinformatics. You need synthetic biology, the tools to move DNA sequences and put them into a strain. And so this is strain construction and growth. This could be one of the limiting factors right now into drug discovery, is a lot of strains we use might not be suitable for certain production of certain molecules. So there are new species or new taxa that are being exploited or being studied in order to find better strains to use in this process. And finally, you need bio and physical chemistry. So you need a multidisciplinary approach in synthetic biology. And of course, this process, this model is repeated, and the intelligent reiterations can go through traditional way, which is humans looking at data and making decisions. Or nowadays, a lot of labs are opting for using machine learning and using an algorithm that will look at the data and will make a decision on what would be the best new round of DBT, which is design, build, and test. As I said, automation is critical nowadays. So this is just one prestigious program that is being developed. It's a master's in automated science that is open. So for students who are interested, this is one reference we could look at if we want to do some of this work later on. Let me just show you a small video of what is the lab of the future look like. So basically you have laboratories that contain several stations with a central robotic arm. And this robotic arm is going to move through different stations: the cloning station, the PCR station, the transformation station, the incubation stations for growing strains, the screening station, colony picking. Everything that traditionally done by hand, with few exceptions, is now being automated. So there is a lot of money being invested in lab automation. And as I said, because they need people who are qualified, there are a lot of programs that are now coming up that deal with lab automation. Machine learning is going to be central. Robotics, machine learning, and artificial intelligence. So data generation, data interpretation, and experimental selection, all of these is going towards more automated approaches and more algorithm-based selection of protocols that would be the best path to follow to make the best strain for producing compound X, Y, or Z. And so this is just my brief overview of this field. I didn't go into any details. There's a lot that we can unpack here, but the time doesn't permit that. So we'll just move into the last part of my talk. And this is something that I would like to share with other instructors like myself to bring research into the classroom and to bring biodiversity and to build banks of strains from small universities. This is a program that I've been using since 2017 in my microbiology lab course, and it's called Tiny Earth. And basically the overall approach is to expose students, especially particularly undergraduate students, to the scientific work. If you know, or you should know, that traditional microbiology labs, what they do is they teach students different techniques with strains that are pre-grown in the lab. And so it's kind of dry. It's not, doesn't feel like the students are involved in something bigger than the classroom. And in this program, allow the students to feel like they are part of a bigger network of other students and other instructors and research, and they feel like you're doing a research project. And they do a research project. They start at the beginning of the semester and they finish it at the end. I can tell you more about this. I'm going to talk a little bit about it, but if you are interested, talk to me, and then I can point you or connect you with the right people that could train you to become involved in this. If you are close by, I can train you myself because that is also a possibility. But let me tell you a little bit more about it. So what is it is a student sourcing. We focus on antibiotic discovery. As I said, there's a crisis in antibiotic discovery. This is a program that was established or is headquartered at the University of Wisconsin-Madison and Wisconsin Institute for Discovery. And as of the latest numbers we have, it was involved in few labs or few schools in the U.S. Now it has expanded. It involves many universities or community colleges or labs. More than 10,000 students that are involved, about 45 states and 23 countries worldwide. As you can see, Morocco is still not on this list. I would love to see Morocco in this list because I really believe that this is a very, very useful program for the classroom, for students. And it's not difficult. That's the other thing. It's not difficult to implement. The training doesn't take that long, and it's very, very simple to implement. So how does it work? Basically, at the beginning of the semester, we ask students to bring a soil sample of their choice. We give them few guidelines on what to avoid, because we don't want them to bring soils that might have pathogens. So we give them a certain guidelines, but they get to choose their own environmental sample. So early on in the program, they feel implicated in this research. So once they choose the soil sample, then they bring it into the classroom, and then we help them isolate the microbes from the soil sample. And then once they isolate the microbes, we teach them how to pick colonies and how to prioritize microbes. Basically choose colonies that look like they have interesting properties. So like, for example, this colony right here, you can see that there is a zone of inhibition around it. So this is a good candidate, but there are other bacteria. So they do a lot of looking at the plate. So it's more, as I said, unlike traditional microbiology labs, there is not just one or two or three strains that are involved. You have a great diversity. And this is also interesting for the students to see that the microbial world is very diverse. So once they select about, let's say, 12 candidates, they put them on a master plate. And then from this master plate, they start to test these colonies against known relatives of what we call escape pathogens. These are microbes with antibacterial resistance. You can see, for example, this is from one of my students. There are several colonies, and you can see that his candidate number 4 and number 5, they do show some inhibition against one of the relatives of we call them safe relatives of one of the superbugs, the highly resistant bacteria. So this is a good candidate. This can then be further characterized. And so then they use traditional microbiology techniques, biochemical techniques, but they also do sequencing. We have them do 16S ribosomal sequencing. Sequencing now is cheap. Once you buy a set of primers, you can use them over several semesters. So you don't have to buy them every semester. And it just doesn't cost that much to sequence the PCR. So they do PCR and they do a lot of cool techniques that they might not be exposed to in traditional labs. And if they find something interesting, then we can find collaboration either within the school or, for example, we can reach out to chemistry departments to extract the antibiotics and test them, purify them, and so on. Or we can reach out to other labs outside. So this is also extremely important because it encourages collaboration. So this tiny Earth network have multiple institutions, multiple facilities with capabilities to do a lot of things. So you don't have to have all the equipment in your own lab or in your small community college. You can reach out to other people in the network to help with these the downstream consequences of the project or downstream uses of the project. So this is one example, a plate coming from a soil sample. This was in 2017. And here you can see this tiny colony with a zone of inhibition. And every time it's interesting, even for the instructor, because it doesn't feel like it's repetitive work. Every time you look at the plates, they are different. They're not exactly the same. So I really enjoy doing this as an instructor, and also the students have more engagement with the course, and overall we see higher success in the classroom or this lab course. Isolation and viable count. So we teach them how to do serial dilutions, and then using serial dilutions, we can isolate the bacteria, then deprioritize the microbes. I'm just gonna skip to this slide. These are some of the slides I use in my course. We teach them how to spot the right colonies and then pick those colonies and transfer them. So they do a lot of microbiology techniques that are relevant either to academia or to the industry. And then test for antibiotic production. So we have them put their candidates against tester strains, different techniques, and then eventually they choose one candidate and they have to maintain a pure culture, amplify the 16S gene for phylogenetic analysis, do microscopic analysis, and do biochemical tests in order to try to characterize the microbe. And this can be done within a traditional semester. It's even been done in an accelerated semester. For example, a summer semester is also possible. Right, so the students get to be exposed to principles such as biosynthesis, the presence of natural products of benefits. They learn about biosynthetic gene clusters, and they finally learn about other techniques besides just the traditional blast or phylogenetic analysis in NCBI. They can go and look at other programs such as anti-SMASH, for example, which is used for discovery of BGCs. This is one example from one of the students. They isolated this bacteria in spring 2020, and we were able to sequence 16S ribosomal RNA. We were able to find that, it was already discovered, but this is a proof of a concept that it works. And we could find that this is a bacteria indeed has the ability to produce a secondary metabolite with activity against bacteria. All right, so I know I rushed through the last part, but that's because of time constraint. So this is my talk, and I hope that you can share thoughts or ideas with me, or if you have questions, please reach out. Before I end, I'd like to thank the organizers for giving me the opportunity to talk to you today. I think this is a great resource for students and instructors alike. I think we have a lot of talent in Morocco, and I think there are a lot of possibilities that can come up from collaboration, and this is one example of collaboration. So thank you, Moulay Abdelmajid, for the opportunity, and I'm going to stop here.
Thank you very much, Tarik, for this outstanding presentation. I enjoyed it very, very much, and I learned also some new things, although I had a master's in microbiology a long time ago, but I learned some new things today. And that's actually the reason we have these seminars, etc., is to encourage our students and faculty, etc., to be here and learn. Yes, so I have a couple of questions, and as I said, you talked a lot about BGCs, and these to me are very similar to operons, like the lac operon, for example, etc. So what are the main differences other than lac operon, for example, are producing the enzymes for the primary metabolism? Are all the BGCs are involved only in secondary metabolites or not?
Not necessarily, but a majority of secondary metabolites are produced by BGCs. But perhaps the major difference is that uh, what's in a lot of operons, the enzymes might not be related. They're not in the same pathway. So like operons, some introduce the compound inside of the cell, but in this biosynthetic gene clusters, they all act on. So it's all a single biosynthetic pathway that will allow the production of a secondary metabolite. And what's really interesting in is not the central genes that make the backbone. It's more those tailoring genes that can add or remove functional groups. And those are actually being explored. And a lot of companies are looking at ways that so they can manipulate their groups. So perhaps I think that's one difference. But also operals all are controlled by a single promoter. Biosynthetic gene clusters are not always like that. So only few are have that central control promoter sequence. So that is also another difference. It's a very good question that I didn't consider like before. When you are inside of it, you don't look at it from the. But
that's fine. That's good. Yeah. And also you talked about some compounds like levastatin and terpenes.
Yeah.
So, and of course you have alkaloids and others. But my question here is about the non-ribosomal peptides. So I've seen at least one there, but through your experience and knowledge, what are the main functions of these? Because, for example, at least in soybean, I know, for example, lunacin is a peptide. It's like a small peptide, 45 amino acids that has tremendous anti-cancer properties.
Yes.
So that's also a secondary metabolite in plants. But what are the main functions in those bacteria of at least these non-ribosomal peptides?
To be honest, I don't have an exhaustive list on top of my mind right now on the different functions, but I have resources that I can look into and get back to you later.
Okay.
I'm not an expert by no means in the secondary metabolism.
Okay.
But there's a lot that I'm learning too. So that's a very good question that I could send you some references that I have here, but I don't on top of my head right now.
Okay, no problem. And I like your project, the project you talked about at the end, Tiny Earth program. And actually I want to implement this in our department. And I want to ask you if you are willing to have a presentation for my department faculty on this, and I will encourage them to actually apply it or bring it to the lab. Right. So if you are willing, let me know. I will schedule that.
I will do it with happily.
Okay.
As I said, it's the more people involved, the bigger the network. As you say, Morocco is not in there, or
I saw that. In Africa, it's only Nigeria, and we can put it there and ask faculty. I will send an email about that to whoever is interested. Can contact you, and we can take it from there.
For more information, there is also a symposium that is organized every year. This year, I am part of the organizing committee, and it's going to be in June. We don't have a final date.
Okay.
But I can send you the information more.
Yeah, please do so. That's great.
So it's going to be virtual.
Thank you again very much, Tarik, and all those who are present. We enjoyed your talk.